So the next speaker is myself. I'm Jake Socha, and the talk title is Does Water Loss Explain Mass Loss During Wing Development in Cicadas? So before I get started, I'd like to acknowledge my co-authors, two undergraduates, Trevor and Alan, who are REU students, as well as their graduate mentor, Josh Taylor, as well. So I think all of you appreciate very well that insect wings are fantastic structures. They're flat plates that enable flight, which is a key innovation for insects that have enabled them to be incredibly speciose and diverse and sort of dominate the earth, of course, right? Insects rule. What's less appreciated is that the insect wing starts out as this little pad that's folded and quite interesting structure, and then fairly rapidly in many, many insects turns into this large wing, which is an incredibly mechanically challenging puzzle. How does this happen? Um, and if you get it wrong, you can get it really wrong, and your insect will not be able to fly. So we know something about this process. So this is from a paper from this year, quite a nice paper. You're looking at a fruit fly on a stick, and a top view and a side view, and what you're going to watch is a time-lapse video of the animal here has just come out of the pupal case and is going to expand its wings. And what is going on here is the animal is swallowing air, it's pressurizing its body, and it is using its circulatory system to blow up its wings. So it's a hydraulic expansion of the wings that turns them into these little beautiful flat plates. In this experiment, they also measured pressure in the body. So this is a pressure probe in the thorax here. And what you're going to watch is the pressure within the body as this process happens. So you can see the wings folded here. The pressure is going to rise. It will soon hit this plateau. And when it hits this plateau of about 3 kilopascals, that is when the wings will expand. Okay, so again, this is a circulatory hydraulic process. But not all insects are fruit flies. And in terms of trying to understand what's going on in these wings, they're pretty small. So we work on bigger animals. This is a nymph of a cicada. And what cicadas do is they spend a lot of time underground. They crawl out. They crawl to something to climb on. They go upward, and then they pop out of their pupal case. They pressurize the body here, and then their wings are expanded. That's a time lapse as well. It takes place over the course of about 30 minutes. So previously, we did a study on these cicadas where we allowed the nymphs to crawl up a wall that was lined with mesh. And what we did is we timed these events, and we pulled the cicadas off at 5-minute intervals, and then we dissected their wings. And our question is, how much of the hemolymph, the blood, actually flows into the wings? What is the mass change? Okay, so all these points that you see here, these 736 wings from 184 cicadas, those are all at these 5-minute intervals. So you see here that the wingspan goes up over the course of about 30 minutes. That's the expansion. That's as expected. So what you're looking at here is what happens to the wing mass over this same time period. And what you see is that there's a massive amount of blood that flows in, roughly changing the mass of the wing by a factor of 3. So that's pretty interesting. But what is perhaps more interesting is that you then see the mass go down, and it goes down fairly rapidly in the course of about an hour and a half or 2 hours. And the mass level goes down to the original mass of the original wing. Okay? So to us, this appears like the blood is being pumped in, and then the blood is being pumped out. Okay? So that's what we think is going on. Okay? But we don't quite know because all we did in this experiment was essentially chop the wings off and measure their mass, right? We can't see what's going on. So what explains this rapid mass loss that we see from this experiment? Now, to understand this, you need to understand what's going on in a wing, which I think the speakers here have explained quite nicely. Really, really beautiful work. This is a wing vein cross-section, and within the cross-section, we have hemolymph, which is flowing through the veins. This is work of my former postdoc, Mary Salcedo, when she was doing her PhD work with Stacy Combs. And you can see here that the hemolymph is flowing through. But also, there's a tracheal tube in many of these veins, okay? So there's a space for air within this tube. So it's a tube within a tube. Okay, so we're trying to figure out what this mass loss is. I'm going to turn this into a bit of a cartoon here. So there's our mass curve. And here are the wings as they expand. And here's the full expansion point. And then a couple hours later, we have this, this nice light wing. So we think that the blood is being pumped out during this phase here. But alternatively, it's quite possible that we simply have water loss across the tissue, right? So this entire mass loss could be water loss. Okay, we can't rule that out. Okay, so that's one of the things that we're going to be addressing in this talk today. Now, there's a mechanical problem with this blood pumping out hypothesis, and here's the problem. So here's the forewing an hour and a half after the full expansion, and it's this light, beautiful wing that is stiff, okay, and it looks like what a wing should look like. Let's go back in time to the point of full expansion. Here's a wing that I'm playing with. I'm trying to actually get a fold out of it, and you see that it's floppy. This thing is wet, it's floppy. I have to use my forceps here to straighten it out on the foil. It is not what you think a wing is whatsoever, okay? That's a pretty massive change, mechanical change in a short amount of time. Okay, so here's the problem. If you pump fluid into this soft structure and then you pump it out, what is going to happen is that soft structure is going to collapse, okay? So, but you saw the wing there, it was not collapsed. So if you're pumping the blood out of this wing, how does that happen, okay, without collapse? Now, what I thought might be happening is the same thing that Wasserthal figured out in the 80s and 90s, which is in this giant atlas moth, if you look in the vein there, you see that in some cases you have a collapsed trachea, in some cases you have an expanded trachea. So that air then can displace the fluid volume in that vein, okay? And Wasserthal actually hypothesized that this is what is happening during wing expansion in this giant atlas moth as well. So not surprisingly, we have a German who explained something decades ago that I'm trying to talk about again today, okay? So, okay, so we did our work on the cicadas that came out this past summer. I'm sure some of you experienced this. It was immense and amazing. The last time these two broods came out at the same time, Jefferson was president. So it was awesome. So what do we do? So the nymphs coming out of these holes, they usually come out in the late afternoon or evening. We collected them like mad. We collected lots of them. And then we put them on a PVC pipe that was wrapped with plastic mesh, allowed them to climb up. And then we simply monitored and let them do their thing of popping out. And then we did two experiments. We did a water loss experiment, and then we did a vein visualization experiment. So in the water loss experiment, what we did is we cut the wings off, and then we sealed them with a small amount of UV activated glue. And then with the wings, we put them on a mass balance, and we weighed them every 5 minutes for 60 minutes. Now, in comparison to watching flying snakes flying through the air, this is literally like watching paint dry. But the data are quite reliable, so that's nice. Okay, so we had 4 stages of wings, and to understand this, I want you to see this nymph here. It has a forewing pad, and it has a hindwing pad, and this is a key to the timing here. So, so if we watch this, this animal emerge again, we see the wing is coming out of its case, and at this point, it will pop out. Boom, it pops out. Okay, so as soon as that pops out, that is defined as time 0. Okay, so we took some of those wings at time 0. We took a different set of wings after the animal has done its thing and it has reached full expansion. And there you are, you're at full expansion. And this should be the massiest wings that we have, that goopy, floppy wing, right? Okay, then we waited for, in another set, we watched them to full expansion, and then we collected the lighter wing. So these are full expansion plus 1.5 hours. And then lastly, just for point of comparison, we also, there are tons of adults around, so we did some adults as well. Okay, so these are the data. How does the water loss rate vary by stage? We care most about this full expansion one to compare to our other experiment. But because we have it, let's do a comparison across these different wing stages. So the first thing to note is that the mass went down over time. That's good. Okay, we can measure mass. The second thing is you have a massive amount of variance here in these time zero wings. And we think that's because when you cut them, there's a lot of hemolymph that comes out, and it's quite variable how much, and it's very hard to control. Okay, so this is experimental slop. Okay. Second point, though, is the slopes are really what matter here. How fast is it losing water? How fast is it drying out? So let's reference everything by these full expansion wings. Okay, so full expansion wings are a reference of 1, of course. If you look at time 0, they lose water about 3 times as fast as the full expansion ones. After an hour and a half, the rate is about 0.8, so slightly slower than those full expansion ones. And the adults then are about 1/10 as lossy. So the tissue is sclerotized there, and they're much better at not drying out. Okay, so here's the actual important plot. Okay, this is the punchline to our story. We're going to compare it to our previous data. So these are the full expansion wings, the water loss rate for those samples. So if we now put the regression line from our 2023 data, what you can see here is that these are quite different. How different, we haven't quantified that yet. But you can see there that there is, the water loss does not fully explain the mass loss that we saw in this other experiment. There is something else that is big that is going on. Okay, so for experiment number 2, we did a visualization of the wings. We have this microscope objective on a camera and a second camera to look at the full wings. So with our microscope objective, we could look at a vein here, and then there's the wing expansion for reference. So in this plot that you're seeing here, we have tracheal diameter in the vein in red, and then we have in black, we have the wing length. And we can see when the wing reaches its full expansion point, so that's here at 100%. And if we now look at the tracheal diameter, what we see, so in going from this point A to this point B, is we see a very rapid expansion of the tracheal tube within the vein there. Okay, so this was pretty exciting for us to see because we could see it directly, and it was very fast. Okay, so on average, this is taking place in about 4 minutes. And it's always occurring before the full expansion. Okay, so this was quite intriguing for us. Okay, so in conclusion, what is explaining the mass loss? Is it water loss? The answer is yes, it is, but that's a minor point. Okay, is the blood being pumped out? We have support for that. We have the tracheal tube that's expanding to take the place of that would prevent the collapse. But we don't have a direct visualization of the hemolymph, as you saw in some of these other talks, which is possible. And what we'd really like to do is take this to a synchrotron and view the tracheal tubes directly, really clearly, and view the hemolymph flow as well to quantify what's going on here. So with that, I'd like to thank the following people, and I'm happy to take questions if there's time, because I have no idea. I have 1 minute. So I'll take a question as we switch over.
I was wondering, but I missed other talks, what makes the wings stiff? Is it the veins or the skin?
Stacy is sitting right there. It's a combination of both, right? But it's probably more so the veins. Yeah. I'm sorry? Well, no, that vein tissue, the wall there is what hardens up. And if you dry the wing out, it has very different mechanical properties. Yeah, that's the key. And that hemolymph there keeps the wing wet and it keeps it with that important flexible property.